Hi folks, my name is Cole, and today on Investigate, Explore, Discover, we're going to be talking about how modified macrophages alleviate Alzheimer's-related disease. Now, Alzheimer's disease fits under the umbrella term of dementia, and dementia is a general term for symptoms like decline in memory, reasoning, or other thinking skills. Worldwide, 50 million people are affected by dementia, and in the population of people over 60, 5 to 8% of them have dementia. Furthermore, this is the fifth leading cause of death worldwide, and Alzheimer's disease makes up over half of the diagnoses of dementia. This here is a map of dementia prevalence worldwide, and you can see that it is localized to lower income countries. Now, with the global population aging, there's actually projections for dementia to increase up until 2050. Dementia is not without costs itself. It costs the world $818 billion per year, which is about 1.1% of GDP. And the majority of care for people suffering from dementia is provided by family carers. And it can be overwhelming for these people. There is physical, emotional, and financial pressures that can cause great strains to families and carers, and support is required from health, social, financial, and legal systems. Now, Alzheimer's disease is characterized by a decline in memory, reasoning, and thinking skills, which may manifest as mood swings and social withdrawal. Just like how dementia is not equally spread out throughout the world, Alzheimer's disease affects women more than men, where 65% of people diagnosed with Alzheimer's disease are women. You may not have encountered someone with Alzheimer's, and so you may be a little unfamiliar with how this looks or feels. This here is a series of self-portraits by William Utermohlen describing how he sees himself as the years progress from his initial diagnoses of Alzheimer's up until when he was admitted into the hospital. Alzheimer's disease is a disease of the brain, and your brain is made up of normally functioning neurons. However, in Alzheimer's disease, there is a presence of amyloid plaques and tau protein tangles, which cause chronic inflammation in the brain, which actually decreases the brain matter as symptoms get worse and the disease progresses. Amyloid beta is actually a naturally occurring protein in the brain. It comes from a protein found on neuronal cells called amyloid precursor protein, and this protein is normally cleaved through normal synaptic transmission. However, about 10% of it gets erroneously cleaved into amyloid beta, and about 5 to 10% of those erroneously cleaved peptides are actually pathogenic, and they form amyloid plaques, which, when built up, inhibit ion channels, calcium homeostasis, and impairs neuronal energy metabolism, eventually leading to neuronal cell death. Because this protein naturally occurs, the body has many ways of getting rid of amyloid beta from neurons. So when found in the brain, they can be enzymatically cleaved, phagocytosed or uptaken by immune cells in the brain. They can diffuse out of the brain or be actively transported out of the brain. Now, there are many therapies for those experiencing Alzheimer's disease, and one of them that is proven recently to be effective in helping people deal with Alzheimer's is humor therapy. The good news is if you don't know a lot of jokes, you only really need one good one. If you don't have one, I'll share one of my favorites with you, which is, when does a joke become a dad joke? That's when it becomes apparent. I know comedy's not my strong suit, so we'll go back to what is, which is immunology. So I mentioned that there are immune cells in the brain, and these are called microglia. They're effectively specialized macrophages in the brain. Throughout our body, though, we have multiple different immune cells. We're going to be focusing in today on some of the innate response cells, which have fast-acting, non-specific responses to pathogens and injury. And these cells that we're going to focus on today are monocytes and macrophages. Monocytes are a cell that are typically found in the blood moving around, and when they get to tissues, they differentiate into macrophages. Macrophages are best known for their ability to uptake foreign bodies, such as harmful proteins or bacteria, and can release cytokines that are either pro-inflammatory in nature, such as TNF-alpha, or anti-inflammatory in nature, such as TGF-beta, IL-10, and IGF-1. To mediate this activity, macrophages have a number of different receptors which help it interact with the environment and recognize what's going on around it. In addition to receptors, they also have different proteins on the cell surface, proteins such as angiotensin converting enzyme, or ACE. Now, ACE is something that converts angiotensin 1 into angiotensin 2, which angiotensin 2 controls blood pressure by causing blood vessels to constrict. ACE is also an amyloid beta degrading enzyme. There has been previous research on macrophages and microglia and their function in Alzheimer's disease. However, the role of each of them is still debated, and ACE, which can be found on these cells, is actually a gene of interest in Alzheimer's disease because overexpression of this protein increases innate immune responses. And in a mouse model, ACE overexpressing monocytes, macrophages, and microglia caused no to little progression of Alzheimer's disease. Even though there has been some research already done on microglia and monocyte ACE overexpression, it's important to continue to look into this. The paper that we're looking at today highlights the role of ACE overexpressing monocytes as opposed to microglia in Alzheimer's prevention. Additionally, this paper investigates the therapeutic potential of periodic treatment with ACE overexpressing monocytes. So this paper is called Peripherally Derived Angiotensin Converting Enzyme-Enhanced Macrophages Alleviate Alzheimer's-Related Disease by Koronyo-Hamaoui et al. From the Cedars-Sinai Medical Center in Los Angeles, California. And how they go about doing their experiments is by utilizing a mouse model of Alzheimer's disease. What they first did is they cleared all of the immune cells in their mice and injected in different types of monocytes, either from an Alzheimer's disease mouse or monocytes from a wild-type mouse and monocytes from a wild-type mouse overexpressing ACE. And they found that after 7 weeks, these cells that were from non-Alzheimer's disease related mice were able to stick around and be present in about 30 to 40% of the monocyte cell population, indicating successful engraftment. And after 7 months, they looked in the brain of these mice and found that when injected with healthy monocytes, there was a decrease in amyloid beta in the brain and a congregation of macrophages around these amyloid beta plaques. Furthermore, they found that ACE overexpressing macrophages were found at about 3 times higher level than the wild types. And these same macrophages had increased anti-inflammatory cytokine production and decreased pro-inflammatory cytokine production. Transplanting in immune cells can be difficult. So the authors of this paper thought of another method of giving these immune cells, and that is through periodic treatments. So they found that when treating with a saline solution control, wild-type monocytes or monocytes overexpressing ACE in their Alzheimer's disease model and compared it to a control mouse. And they looked at a bunch of different cognitive tests that they could administer to these mice. And they found that, unsurprisingly, the control mouse worked the best, but when their Alzheimer's mice treated with ACE overexpressing monocytes were compared to the control, they found that their cognitive results were very similar, which is very promising. Furthermore, they looked at the monocytes directly and found, compared to wild-type macrophages, which when stimulated with amyloid beta produce a lot of pro-inflammatory cytokines, ACE overexpressing macrophages still had an increase in pro-inflammatory cytokine release, but this increase was much smaller than the wild-type. Additionally, the ACE overexpressing monocytes had an elongated phenotype, which is known to show anti-inflammatory and pro-healing qualities. So to see how exactly their macrophages were interacting with amyloid beta, the authors looked at the macrophages themselves and found that they had increased expression of membrane scavenger receptors, which have been implicated in macrophage activation and increased phagocytosis of amyloid beta. And to see whether ACE was responsible for this phenotype, they blocked ACE activity and found that the presence of ACE is what makes these cells uptake amyloid beta more. So to quantitatively investigate how ACE overexpressing monocytes function, they exposed these cells to a bunch of amyloid beta and tested their ability to clear amyloid beta from the media that these cells were in. Yeah, and they found that ACE overexpressing monocytes actually decrease the general concentration of amyloid beta over time and have an increased amount of amyloid beta inside of the cells, which is super cool and illustrates that ACE overexpressing monocytes enhance degradation of amyloid beta. This information is significant and contributes to our overall knowledge in some pretty cool ways. It provides a measurable account of amyloid beta clearance by ACE overexpressing macrophages and shows the role of ACE monocytes and macrophages specifically to clear amyloid beta plaques by themselves. This then illustrates that ACE overexpressing monocytes and macrophages can be possibly used for future therapy for Alzheimer's disease. There's a lot to go through for this paper, and I had some questions when going through it. Firstly, because ACE overexpressing monocytes and macrophages can clear amyloid beta, can they also clear tau tangles, which are found in Alzheimer's disease patients? Furthermore, we have medication right now called ACE inhibitors, which prevent the activity of ACE to reduce high blood pressure and heart disease. So what are the possible adverse side effects of having cells overexpress ACE? There is previous research on ACE overexpressing monocytes and macrophages. These cells have been shown to help alleviate melanoma and to help clear acute foreign invasions by pathogens. So what other diseases can these macrophages be used for? Can they be used for different types of cancer? Can they be used for autoimmune diseases? I don't know. But what I do know is that I want to hear what you think. What are some of the questions that this information caused you to think about? I would also like to hear if you are interested in any specific topics and what you would like to hear about in the future. Let me know about those in the comments below. But otherwise, I really hope that you learned something. And more importantly, you had fun doing so. So if you did, give this video a like and subscribe for more in the future. And with that, I will see you next time.